Welcome to the deep dive. Today we're looking at something, well, really fascinating coming out of USC. It's a new angle on cancer immunotherapy.
That's right. It looks like it could potentially be a real game changer, especially for those solid tumors that are notoriously difficult to treat.
Yeah, exactly. We've been digging into the research you sent over, listener, about these EchoBack CAR T-cells. So our aim today is to really unpack what makes this approach so different and, well, potentially significant.
Okay, so the core idea, fundamentally, is using CAR T-cells, those engineered immune cells, but adding a new layer of control.
Control using focused ultrasound.
Precisely. Focused ultrasound to sort of guide and activate them, making them attack tumors more persistently.
And the potential upsides seem pretty clear right off the bat. Longer effectiveness, like over days,
Right.
just hours, and targeted activation just
Yeah.
near the cancer, which, could mean fewer hospital visits, maybe better outcomes overall.
That's definitely the hope. And it builds on existing CAR T-cell therapy, which has already had some, real successes.
Right, especially in blood cancers like leukemia and lymphoma.
Exactly. So maybe we should just quickly recap what standard CAR T-cell therapy is.
Good idea. So basically, you take a patient's own T cells, right? They're immune warrior cells, and you engineer them in the lab to specifically recognize and hunt down cancer cells. And Professor Peter Yingxiao Wang at USC, he's been a big name in this field.
A leading figure, definitely.
Yeah.
But translating that success from blood cancers over to solid tumors, that's been the big hurdle.
Okay, so let's start there. Why are solid tumors so much harder to tackle with these kinds of immunotherapies?
Well, it's a few things, really. Solid tumors often build physical barriers. They could literally block immune cells from getting in.
Like a fortress wall.
Sort of, yeah.
Yeah.
And inside, they create this microenvironment. It's actively immunosuppressive. It basically tells the T cells to, calm down, stop attacking.
Wow. Okay, so defenses and internal sabotage?
Pretty much. Plus, they're often just complex structures, not uniformly spread like blood cancers can be. Makes a coordinated attack harder.
Right. So how do these new EchoBack CAR T-cells plan to sort of outsmart these defenses?
Well, a big part of it is this improved control and, crucially, the duration of the attack.
Okay.
You see, standard CAR T-cells and even some of the earlier attempts using ultrasound, they had limits on how long they could keep fighting effectively.
I think the research mentioned something like maybe only 24 hours of activity for some earlier ultrasound types.
That's right. But these EchoBack cells, the data suggests they might keep attacking for up to 5 times longer. We're talking days, not just one day.
Okay, that is a big jump. Days versus hours. That sounds like it could make a real therapeutic difference.
It really could. And being able to remotely switch them on or prime them with a focused ultrasound, that's key for hitting the tumor hard while hopefully leaving healthy tissue alone. Better safety.
Right, hitting only where you need to. And Longwei Liu, the lead author, mentioned this could really cut down on how often patients need treatment, right? Like maybe only once every couple of weeks.
Yeah, potentially even less frequent. Imagine going from frequent infusions to maybe just a couple of visits a month. That's a huge quality of life improvement.
Absolutely. You can see why Professor Wang called it a breakthrough for making this kind of ultrasound control actually practical for, real medicine.
It bridges that gap from lab concept to potential clinical reality.
Okay, so let's get into the echo part. The focused ultrasound is the on switch. How does that work exactly? Just a zap of energy?
It's a bit more precise than a zap. It's a focused pulse, relatively short, maybe 10 minutes.
Okay.
And this pulse triggers something inside the EchoBack cells. It primes them, makes them better at sensing nearby cancer cells, like a targeted wake-up call right at the tumor site.
So a quick burst of ultrasound focuses their attention. And Professor Wang highlighted that this leads to better local tumor killing, a milestone, he called it. But the effect lasts after the ultrasound stops.
Exactly. The ultrasound pulse itself is brief, but the anti-cancer activity it kicks off is sustained. And that's where this EchoBack name comes from. It hints at this unique feedback mechanism. What did you make of that?
Yeah, this part is really interesting. They call them EchoBack CAR because the initial ultrasound stimulation gets, echoed, amplified somehow. It's described as a call-and-response-like feedback function.
Okay, a call and response between which cells?
Between the EchoBack CAR T-cell and the tumor cell itself. So Longwei Liu explains it like this. The CAR T-cell finds a tumor cell, and the tumor cell basically sends a signal back to the CAR T-cell.
Okay, and that's signaled as what?
It triggers the CAR T-cell to produce more of its cancer-killing molecules. It's like the tumor accidentally tells the T-cell, hey, attack me harder.
That's so good. Quite a clever biological loop. The cancer fuels its own destruction in a way.
Seems like it. And this feedback loop is also part of the safety mechanism, isn't it?
Absolutely. Because as Lou points out, these are smart CAR T-cells when they drift away from the tumor.
Outside that immediate signal range.
Right. The engineered part, the CAR molecule that lets them see the cancer, it starts to naturally break down, degrade.
So they stop being attack cells when they're near healthy tissue.
Exactly. It prevents them from causing damage elsewhere. That localized self-regulating activity is really promising.
Okay, so you have the ultrasound for the initial kickstart in the right place.
Targeted activation.
And then this feedback loop with the tumor cells keeps the attack going strong, but only locally. That sounds pretty elegant.
It is. It addresses both efficacy and safety. So how did this play out in the lab? What did the mouse model experiments show?
Well, they tested these EchoBack cells against a couple of different human tumor types grown in mice, specifically prostate cancer and glioblastoma cells.
Both pretty challenging cancers, and the results look good.
They looked quite positive, yeah. Lou highlighted that mice getting the ultrasound-controllable CAR T-cells, especially with 2 rounds of that ultrasound stimulation, showed significantly better results, better tumor control, compared to mice getting just standard CAR T-cells without the ultrasound boost.
And did they address that exhaustion problem you mentioned earlier, where CAR T-cells can sort of run out of steam?
They did. That was a key finding. When they continuously challenged the cells with tumor cells in the lab.
Simulating that tough tumor environment.
Exactly. The EchoBack CAR T-cells showed, well, better function over time. They seemed less exhausted and kept killing tumor cells more effectively than the standard ones. That sustained fight is crucial.
It really is. And, it's worth mentioning, this was a big team effort.
Oh, absolutely. The paper mentions PhD students Pixiang, He, and Yixuan Wang doing significant work, plus collaborations with Yale and UNC Chapel Hill for some advanced genetic analysis.
Single-cell sequencing, yeah. Very sophisticated stuff.
And Professor Kifa Zhu bringing in the ultrasound tech expertise. It really takes a village, doesn't it?
It often does for this kind of complex interdisciplinary research. Pushing boundaries requires combining different skill sets.
So, thinking big picture now, what could this mean for the future, if this pans out in humans?
Well, look, it's still early days. Moving from mice to people is a big step.
Yeah.
But if it translates, it represents a move towards potentially more powerful, much more precise.
And hopefully more patient-friendly.
Exactly. Treatments for these solid tumors that have just been so resistant.
Longwei Lu's quote really sticks out, calling it a real step toward the future of safe and efficient immunotherapy for these difficult cancers. That sounds like genuine optimism.
And it might not just be for prostate cancer and glioblastoma.
Yeah.
They seem hopeful this could be a kind of modular tool.
Meaning adaptable to other cancers.
Potentially, yes.
Yeah.
Things like breast cancer, maybe retinoblastoma. If the core mechanism can be adapted, that obviously broadens the impact significantly.
That adaptability would be huge. And Lu seemed really excited about just how smart these cells are designed to be. It's not just brute force killing, it's controlled, responsive.
That's the goal.
Yeah.
Smarter, safer, more effective. It really could shift the paradigm for solid tumor immunotherapy.
And for anyone listening who wants to read the original research, it was published in Cell, April 2nd, 2025. Lead author Longwei Lu, senior author Yingxiao Wang.
Right. And we should probably mention the DOI for easy finding.
Yeah.
10.1016J.cell.2025.02.035.
Perfect. So wrapping this up, our deep dive today really highlights EchoBack CAR T-cells as a potentially major step forward. This combination of engineered T-cells, focused ultrasound, and this clever feedback loop. It offers a way to attack solid tumors that seems more targeted, much more sustained, and possibly safer too.
And the downstream effects for patients could be really substantial. Fewer hospital trips, maybe fewer difficult side effects, and ultimately the hope for better success against some really tough cancers.
Which leaves us with a final thought for you, our listener. If we can create remotely controlled, smart immune cells like this for cancer, what else could this kind of precise, adaptable therapy be used for? Beyond cancer, what other diseases might benefit from this level of biological control? Something to ponder. Thanks for joining us for this deep dive.